Hello everyone. It's Russ of Aquarimax, and I'm here with Kyle Candillian from Roach Crossing. Welcome, Kyle.
Hello. Thank you for having me once again.
Yeah, great to have you as always. We've talked about pseudoscorpions. We've talked about actually a lot of different things on the other episodes that we've done. And today we have something that I've been long fascinated with, and you've had a lot of success with, communal enclosures of multi-species, both in terms of roaches, isopods. You've probably done it with some other invertebrates too as well, I would imagine. So that's what today's episode is about. We're really excited about it. Kyle has brought some critters to show us as well as part of that. So just so everybody knows, that's the focus of today, multi-species enclosures of invertebrates. Very excited about it. So Kyle, tell us a little bit about, just to introduce this topic, how long have you been doing like multi-species roaches, multi-species isopods, that kind of thing?
So intentionally, it's been far more recent. Accidentally goes back to beginning to keep roaches. I'm sure there are plenty of people who've had a couple of different roach species and a couple have gotten into an enclosure accidentally, or maybe you live somewhere like Florida where there are just wild roaches of all different sorts roaming around, and you happen to have something get into your bin and then it starts reproducing with your feeder roaches. So I've had different multi-species communals, pretty much since I started keeping roaches. Only recently, like I said, has it been more intentional, as having proper barriers and better means of securing small climbing species and whatnot have become available.
Okay.
So,
and how long would you say you've been doing it intentionally then? Approximately.
Maybe about 6 years-ish in one way or another. 5-6 years.
Okay, so still a decent amount of time.
Mm-hmm.
Long enough to prove that it works long term.
Mm-hmm.
Yeah, cool. That's awesome. What about the isopods? So I know that almost everyone who's kept any number of isopods has unintentionally kept dwarf whites with something else, at least one other species, and that can end badly. But what about intentional success with isopods that you've had multi-species?
So most of my isopods are kept multi-species. This is everything from the larger Spanish Porcelio to the Nasodillo cubaris, etc. And I found that, depending on the mixture of species and, the stock that you receive, you can get good results, you can get poor results. So it varies, depending on your combination and your husbandry and whatnot.
Yeah, I find this really fascinating because early on in my isopod hobby, I found, I had some of those bad results. And and then even later on with some of my experiments, I've had bad results. Like when I tried to keep zebras with powder blues in a bioactive enclosure, it worked for about 1.5 years. And then boom, zebras died out and the powder blues are still going strong 3 or 4 years later. So that's this idea of having certain combinations that work really well fascinates me. So I want to make sure we definitely give plenty of coverage to both the roaches and the isopods this evening. Where would you like to start?
I think I'd like to start on sort of the basis to some of this because there's both the natural perspective and then the hobbyist perspective on these sort of things. So if we look into natural roach and isopod communities, and this applies more directly to isopods, when everybody's lifted up a board and saw, just hordes of some sort of isopod falling off of it outside. If you live in a moist climate, you've had that experience. And it's usually not a monoculture. Usually there's an assemblage of species, and there's a couple that are more prominent than others. And so in the wild, we tend to take things into captivity and we keep them relatively sterile, and we think, we have to focus on this species needs or something like that. And kind of forgetting that, a lot of things go hand in hand and occur in sort of cohorts or groups or, species assemblages in the wild. And that goes not just for, introduced species, but also for native species of different, insects of any sort. So looking at, the isopods in the wild, at least in Michigan, you'll find vulgare, you'll find Porcellio scaber, you'll find Trichelopus rachii, you may get lucky and find some Haplophthalmus danicus. You got a good mix of about 12 different species. And the primary things that seem to be dictating who's where are moisture, soil conditions, and of course the types of leaf litter that are found in that location. And I actually studied this extensively. It was my undergraduate project when I was at U of M Dearborn, was to study the composition of different leaf litter invertebrates in high quality native habitat and in degraded, heavily invaded by different invasive species of plant habitat. And so that sort of has shaped my perceptions on how we keep these different things. Another thing is when we take things out of the wild and bring them into captivity and get a culture going, there is a gigantic thumb on a lot of things in the wild in the form of selective pressure, ecological pressure, based on the moisture and what's available in terms of food, the temperature, wind, etc. All these things that can push down on a species that isn't exactly well adapted to whatever conditions are there. And then, of course, when you bring things into captivity, you take those pressures off. There's nothing competing with them. You're feeding them tons of food. Temperature could be very stable or something like that. Some of these species that don't seem to be abundant in the wild are not very common. All of a sudden, you can produce scads and scads of them in captivity. So working off of that, as I said before, with isopods, usually found with multiple species in the wild, and there's something to that too, because they can't all necessarily be feeding on the same thing or using up the same space. That competition between different species is not good for either species. Actually, competition is bad for both species that partake. It's better for them to find their little niches therein.
Some resource partitioning.
Yes, resource partitioning. So with things like roaches, it's very interesting, and I may make some bold and not necessarily directly founded claims here, but because roaches are such generalists in terms of their dietary needs, with a couple of exceptions, things like Panesthea or Cryptocargus and whatnot, things that are wood eaters or specifically found within ant nests or something like that. They're very generalist. So one of the big thumbs that's usually on them is predation and then availability of things like moisture. So you look to like Florida, where there's just tons and tons of introduced roach species, and you can flip a board and you can find Parcoblatta fulvescens living with Epilampromaya, living with Ischnoptera bilunata. You can just find all these roaches in the same place at the same time using this resource. And I believe that's because they are generalists and they are very specialized in what they do, being detritivores that can recycle a lot of their nitrogen using their symbiotic bladybacterium. So in terms of, it's more a matter of finding, physical space that they can inhabit, and then, whether or not there's going to be something eating them there. So a lot of them can be kept together with the same food, pretty much the same conditions with a couple of caveats. And although there are roaches that, seem to need more specialized care in captivity, some things need more heat or some things need to be wood feeding, you can keep them with a lot of other roaches because they don't necessarily recognize each other as a predator or as prey. There may be some overlapping in the pheromones or the similarity of pheromones there, which makes a lot of them very comfortable with each other. Mm-hmm. So an example that has happened a couple of times when I was living at my parents', I had some roaches get out. I think it was probably Shelfridella lateralis and maybe some lobster roaches or something like that at some point. And there's something that I left on the ground in the basement off in the corner, in the moist corner of the basement over by the washing machine. And I went to move it, and all these roaches had run out from underneath it. It was like, why didn't they why aren't they scattered around the room? Why aren't there some of them that decided to hide under the laundry machine? Why did all of them congregate, under this one particular piece? And I think it's because there's probably an overlap in some of those pheromones, and also because, this may have been the most appealing, most perfect moisture, whatever piece of laundry that was on the floor that they all decided to use. Some species have a natural tendency to group up with other species. It's kind of like with birds, where you can see in migratory flocks different finches, and even different, entirely different families or subfamilies of birds in the same flock at migration. there's not really a lot of there's not the pressure on them to compete for resources. It's more about being able to move as a group and migrate. And so they just sort of hang out together, and that's fine with them.
Mhm.
No aggression or anything like that. So, so building off of that is basically there's an inclination, even, in places where there is some sort of pressure on these organisms to share the same space and possibly share the same resources and whatnot, at least for, temporary periods of time. And so in captivity, again, when you take that thumb of ecological pressure on things in terms of resources and space and whatnot, then you can have happy cultures of different types of roaches together, or roaches and isopods, roaches and crickets, all these different combinations.
Mhm.
So
Cool. That's very interesting. The fact that you mentioned the pheromones, that they have compatible pheromones in many cases, and that's probably true with isopods as well. Isopod and isopod, since we know they have, for example, the aggregating pheromone that they release with their frass and so on. That a similar scenario could occur, both in the wild and in captivity. Interesting.
So, and it's we tend to think of things in nature as being sort of perfect and flawless and ruthlessly honed, but the fact is that a lot of these things, they're just, the organism produce these chemicals because they work to do a certain thing, and if it's imperfect, if it can be mimicked by something else, it just happens. So the example I'm aiming for here is, in terms of two different unrelated roaches responding similarly to things that are very similar pheromones. If you take a blue, and it has to be blue, I don't know why, probably something in the dye or whatever, a blue Bic ballpoint pen, and you draw a line on a piece of paper, and you put a reticular termes termite on that line, it will follow the line because the chemical in the Bic pen is similar enough to their trail marking pheromone that they recognize it and they follow it. So, things that are very closely related, in this case, something like two different roaches, even if they're across families, apparently, they may have similar components of their aggregating pheromones that they'll recognize and they'll group up together like that.
Right. And there can be some molecular differences as long as the receptors are the same or close enough. Yeah, that makes a lot of sense. I hadn't thought about it that way. That's a great thing to consider. I like that. Cool. So when you do this, I know that with when you're talking about the thumb, I think that's a great analogy for the selective pressures, this thumb down on the organisms as different biotic, abiotic factors, whatnot. Now, when you have a captive enclosure and the population pressure builds, you're going to have more difficulties, right? I mean, that's just kind of a given. As the population of both species increases and you're getting close to carrying capacity, would you agree that almost always, if assuming you reach that, would you agree that causes more difficulties for them coexisting?
It depends on what exactly there. I think it's mostly based on spatial and on behavioral things in terms of whether or not you're going to have problems with two things at high densities. For example, you can have a colony of hissing cockroaches and have just trillions of dwarf white isopods crawling over every inch of the substrate. But there's not going to be any sort of problems for the hissing cockroaches because, the ooths are their live bearers. So there's not going to be any chances of the dwarf whites eating the ooths. You might have a couple of them try and crawl up on them, but for the most part, the roaches will be more up in the drier parts of the enclosure, and the dwarf whites will be all the way at the bottom. So the chances of there being negative interactions between the two of them very low. Life cycles, there's not really a vulnerable point in the hissing cockroach's life cycle in that scenario. So you're not going to have, even molting isopods, unless they are very, very, very strapped for moisture, they tend not to go after molting roaches. So in a situation like that, as long as, of course, the moisture is kept up and there's enough food for everybody, you won't have any problems with the two of them eating each other or causing problems for each other. And again, part of that's the spatial aspect of and the behavioral parts of their biology.
Right. And part of that being the resource partitioning we were talking about before, and part of it being, like you said, no vulnerabilities in the life cycle of the roaches. And with most isopod species and dwarf whites, the monkai are the vulnerable point. And so dwarf whites and other isopods are going to do much more poorly together than the hissing cockroaches and the dwarf whites. And any two similar isopod species that have basically the same, there's no resource partitioning going on or very little, I would expect those to be the worst candidates for cohabitation.
Yeah, so you wouldn't want to keep dwarf whites and dwarf purples together if you were trying to produce large numbers of both of them, because the dwarf whites will win that any day. So, and yeah, even though they're very similar and fast breeding and whatnot, it's one of those situations where these two organisms have sort of converged on the same niche, and they're from different places. And the thing that allows the dwarf purples to continue existing and doing so well, I would presume, in their natural habitat, is because there are no dwarf whites there, and there never were any dwarf whites there. So it kind of ties into invasive species theory, where if you have something that comes in and puts a lot of pressure. A perfect example would be in Florida, the house geckos. How since about 2008, Hemidactylus maboya has almost completely annihilated all the other species of house geckos in Florida. And they've just completely pushed them out of that niche. So before it was, I think it was the Asian house gecko and another species, Hemidactylus tursicus.
Yeah.
So you may even remember a while ago, you could go into a Petco or a PetSmart and buy a house gecko, and they would have the Hemidactylus tursicus. You don't see that anymore because they've been completely pushed out, just completely eradicated by the Hemidactylus maboya now. And this has been documented. I think in 2017, there was a paper documenting that this had happened. So, two very similar species, and yet unlike, a lot of other invasives in Florida, they don't play nice. They probably directly compete for that same niche of space and resources. And I would assume the maboya probably physically chase out the other species of Hemidactylus. So that's a case where, they're not going to work. But you still have Hemidactylus cohabitating with things like tokays and day geckos in Florida because, they're on different shifts, or they're eating different size prey, or they're avoiding each other, or what have you. So
Right. That's a great example. It reminds me of the one that I would see in Hawaii a lot when I lived there. They had the Anolis carolinensis is invasive there everywhere. It was introduced, I think, in the 50s and did really well by itself. Would colonize the plant everywhere from the ground level up to about 12, 15 feet, something like that. And then in the 80s, I think it was, they introduced the brown anoles, like the ones in Florida that have also been introduced there, same species. And now the brown anoles are much more common, and they tend to dominate the area from ground level up about 3 to 5 feet, something like that. And the green anoles are the ones that are higher up.
Yeah, I think that's going on in Florida also, and that also at the green anoles are still common and still at the northernmost end of the range because of their cold tolerance, and that's basically the thing that gives them the leg up over the brown and Cuban anoles in the southeastern United States. So that's an example where they still persist together. They're not directly, but for whatever reason, behaviorally, they're antagonistic to each other. They're in direct competition or whatever. So, I mean, you can go to Florida and you can see anoles in the same space as brown basilisks or the butterfly or red-headed gammas. And that's because they don't have any antagonistic behaviors to things that look like that. And also they're not directly, competing for either the same space or the same activity period or whatnot.
Right. That makes a lot of sense. Yeah, and I appreciate you saying anoles. I out of habit, I still say anoles, even though
Yeah, I did too. I took a lot of manual effort to change it, and I feel like I just did it because it's funner to say. The E on the end I feel is funner to say. That's the thing.
It is more fun to say. Yeah, I need to get into that
More
habit.
more fun to say.
Yeah, exactly.
More fun. So,
oh, that's cool. Good examples there. So, as far as your roaches, you said you had some there for us to check out, and we can.
I do. I have a couple of species that are prime candidates for cohabitating, not just with other roaches, but with other organisms. And sometimes that happens, as I said, intentionally, sometimes accidentally. Let me bring out this cute girl first. So this is a Uricotis Fluorodona. This is the big torch key color form. So Uricotis in the wild, they're found under various bark and logs and whatnot, usually found with things like click beetles, sometimes with isopods, sometimes with stick bugs. It's a whole bunch of, sometimes with millipedes as well, depending on the age and stage of rot of the wood. And so these Uricotis are an excellent candidate for communal enclosures because they tend to stay up in the drier parts of the enclosure, higher up on the bark and various hides and whatnot. So they don't like to be touching moist substrate constantly. They like to get up to somewhere to sort of dry out a bit. So they're an excellent candidate for keeping with not just other roaches, but with isopods because they rain down poop on the isopods constantly. And the bacterial colonization of their frass is exceptional, perhaps unparalleled isopod food.
Cool. I remember hearing about that can be a great source of isopod food. But do you find that you need to feed the isopods much in addition to that frass that the roaches produce?
So in these enclosures, my primary concern is the roaches, and the isopods are usually the afterthought. And so and everything here gets pretty much everything here gets the same food, the crappiest dog food that money can buy that I would not feed to my actual dog from Menards, and some sort of fruit or vegetable, usually apples or eggplants or squash, or very rarely bananas because of the fruit fly risk. So I don't really think about the isopods, but a lot of the colonies that are kept, most of the colonies kept in with roaches do far better than those that are kept solo in general.
Okay. And what kind of isopods do you keep with this particular roach species?
So these ones with Uricotis Fluorodona, I have kept Porcellio scaber, I have kept Porcellio ornatus, and actually I've kept the regular duckies with them as well. And I've actually been seeding a lot of bins with regular duckies because they seem to do extremely well with roaches. So
interesting.
So the thing with isopod diet is, there's all this, protein, this, calcium. There's calcium in a lot of places you would never expect, including leaf litter and roach frass and whatnot. And what isopods are mostly going after, and there are probably some exceptions with more specialized dietary needs, but what they're primarily going after as detritivores is actually the bacterial colonies that are in the food items that they're going after, the leaves or the rotting wood or the fungi, etc. They're going after the bacteria that are actively breaking those things down. Think of them kind of like cows, how, we see cows eating grass and whatnot. You think, oh, yeah, cows eat the grass. But then you look at cows somewhere like Arizona and you think, well, everything's like dead and dry here. What are they eating? What's actually going on with the cows and why their biology is so weird with all their chambers in their stomach is they're fermenting the grass to produce the bacteria that they then digest, which is why they're able to survive in these places that have suboptimal conditions, is because of that bacteria assemblage. So in a way, they're a lot like isopods and roaches, where the microbiome inside them and the microbiome they're trying to consume is extremely important to their well-being.
That's an excellent point, and I think that's one of the maybe under-discussed reasons why, when you set up isopods, it's a really good idea to inoculate it with some bacteria. You can take a pile of a handful or two of frass from another isopod enclosure and things like that, and get that going first, and that helps you so you're not starving your isopods for the first few weeks until those are set up. So
yeah,
great point.
So things like this and like with that, making sure your leaves and wood are in a proper state of decomposition. I think that once the wood has been colonized by the white rot fungus, isopods are inclined to eat it because the cellulose has become a more digestible, processable form. But actively decomposing wood is very good for a lot of detritivores because it's full of mycelium, which has a lot of nitrogen in it, relative to just the wood that they're consuming itself.
Right.
So thinking about the microbiome in our enclosures, people will set up a bin of isopods or even roaches, and it'll be relatively sterile, and they'll buy a batch from an expo or something that had something like sphagnum moss or paper towel in it. Throw the isopods in the enclosure, and especially if there's something like a wild-caught nasodillo or cubaris, a couple weeks pass, they'll lose a few more. Maybe they bought 10 of them, and then they're left with 2 or 3, and then they'll be waiting months, etc. Maybe they'll get a crop of babies, and then 2 years out, maybe they'll have a healthy colony of some things. And that's because it took all that time to get the momentum built up from the isopods producing frass and the aggregation pheromones and whatnot to make them very comfortable in that bin. And sometimes they don't even get that far. Offering things like live oak leaves that haven't been soaked or decomposed properly, a lot of those things are very unpalatable to these organisms.
Right.
And they have to have special adaptations if they want to feed on things that are not very well decomposed. Things like rhino roaches, where they will take and eat fresh leaves because they're adapted. Fresh, dry, brown leaves, not fresh off the eucalyptus tree in the wild. They're the ones that have fallen and have dried up. In part because
yet.
Yeah, they're adapted to doing that, so they can eat both the ones that have started to decompose as well as the fresh ones that are dead, dry, and brown on the ground. But other roaches and a lot of isopods, people may notice if you put a handful of like fresh off the tree, at this time of year, fallen leaves, and with the isopods, they won't touch them. Things like oak leaves, because the tannins haven't been leached out of them or broken down yet, versus things like maple or elm or mulberry, where it's mostly things like saponins that are protecting the leaves. And those break down very quickly when they're not attached to the tree. Those leaves are a lot more palatable directly to them.
Mm-hmm. Yeah, that makes a lot of sense. And I think when was that? Recently, I was talking to somebody about introducing mycorrhizal starts into a culture to help get that process going. And it seems like it can only help.
Mm-hmm. Well, in my experience, there are some fungi that are not preferential to different captive invertebrates, or at least not generally. Things like the flower pot fungus, which usually pops up in coconut fiber, in my experience, tends to not be very palatable to roaches or isopods for some reason.
That's the one with the yellow. Is that the
yellow?
Yeah,
with the yellow mushroom that comes out of it. So I found that to be not very palatable in general to a lot of things. Versus something like the inky caps, which usually they'll sort of explode into like a dusty black gooey residue on the sides of the tank. Those seem to be a bit more palatable in terms of their mycelium, which are usually colonizing leaf litter or manure in the substrate.
Makes sense. Oh, we've got a question from a comment from 503 Manasha. Yeah, so that's an excellent explanation of why that happens. Maple leaves are probably more saponin rich and not so many tannins, right?
Mm-hmm. Versus oak leaves, which are very rich in tannins. But at the same time, there's more nutrition in the oak leaf that needs to be properly processed to be accessed, because the oaks put there's a lot more resource investment in their leaves as a general trend, which is why a lot of them hang on to their leaves very long into the fall, because they're trying to get the most photosynthesis out of these leaves that they didn't produce until maybe May or June in some places that oaks will leaf out. And then their big, thick, sturdy leaves that are full of tannins that aren't meant to be eaten, because the oaks, they're investing in those to be able to maximize photosynthesis and whatnot, versus something like a mulberry where you can basically hack the wood back constantly for years and you'll keep getting sprouts and whatnot. There's very minimal investment in the wood and in the leaves. Something like a mulberry, saponins are their way of cheaply investing versus tannins. So the type of leaf matters, the age of the leaf, whether or not the leaf was fresh off the tree, whether or not it was a leaf from the fall after various other phytochemicals have been drawn out, and the chlorophyll has also been drawn out of them. So, a lot of things to think about when, a lot of guys will say, feed them leaf litters. That can mean a lot of things,
True.
So.
True. And that's one reason why I like to cover my bases and mix it with lots of different leaf types, because then, you're going to get a variety of composition speeds and a variety of different nutrients and whatnot.
Mm-hmm.
Oh, here's another excellent question from Eileen. Is that showing up on your screen as well?
Mm-hmm.
Okay, excellent.
So occasionally getting gray mites. Usually gray mites are a sign of overfeeding and also high air humidity. So I have occasionally, because I keep a lot of grain pests because I'm crazy, things like the flower beetles and the spider beetles and rice weevils and things like that. Usually there'll be a little boom of gray mites, even if I haven't watered any enclosures in the spring or summer, just because in the bug room, the humidity goes up very gradually and it stays pretty high into the summer. And so I usually have no grain mite issues all winter time because of how dry it is. They can't disperse when it's dry. By the time they've made it halfway between containers, they've desiccated. So that's another factor I would say in there. So cutting back on feeding or or leaving food in for long periods of time is a good way to quickly combat that. Switching the colony to feeding on leaf litter or rotting wood or something else for some period of time that's less palatable to the grain mites. So they're really after those high quality flowery foods. So I'll see it pop up on dog food. Not too often on fish flakes, actually. I think that's because of there's not much grain in there. Things like brewer's yeast and the brine shrimp and stuff like that is not as palatable to the grain mites, but definitely on dog food and things like that. I cannot speak for commercial isopod diets because I have never used them, so I'm not sure what their contents are, but I would assume that if there's a lot of them on the food, it's probably due to there being a lot of grain products mixed in, which is fine. Isopods in general is tridivores for the most part, so
they can thrive on a lot of different ingredients.
Mm-hmm.
There's another good one.
Yeah, so boiling them will leach tannins out very quickly, which is excellent. What I do is I have large trash cans that I leave outside. I fill them with oak leaves in the fall, and then I completely fill the trash cans with water and leave them out there for until I need them. Usually after about 4 months, a lot of the tannins are out, and depending on the temperature outside, they'll begin to ferment, which is what I really want. I tell people that if you take a handful of the leaves that you've been soaking and you smell it and it smells like horse butt, that's the perfect stage of decomposition. So if it smells like a barn, it's in a good state of decomposition to add, because it's been that smell is obviously it's all the bacteria that are breaking down the leaf litter, mostly on the surface, but also in and through the leaves too, depending on how long they've been soaked and whatnot. So it is interesting because people want things. This has been something that's been in the invertebrate hobby for a while, but people want things to be very clean and tidy, and they want to have a method for disposing of frass and waste, and worried about, this is accumulating, what do I do? But some of that messiness, things like the well-rotted leaves, are very important to getting different species to propagate consistently, or just to the general health to make sure that the leaves are palatable and whatnot. That's what I would say. If you boil them to get the tannins out, or just leach them, if you have the time to set up a big bin somewhere to let your leaves soak, just do that. And again, the best thing you can do is just sort of sit on your hands and wait after you've set it up and wait for things to start fermenting. The only thing to watch out for when doing that is depending on what the state of the leaves are when you get them and where you collect them from. If you collect them off of a lawn and you've got, just basically just the pure leaves, you don't have to worry about things like centipedes or earwigs getting in with them, or different species of isopods too. But if you've collected it from somewhere where there's more, mulch or dirt underlaying it, you can scrape up eggs or individuals of those. And some things like the soil centipedes can last a long time in even very, very wet conditions. Other isopods too can last a long period of time, even if the leaves are completely soaked, depending on what type. I think Hyloniscus and Trichoniscus, which people don't seem to keep, probably because they have specialized microbial needs in their food. They're usually found in river corridors with a lot of leaf litter. But those can both survive underwater. They're terrestrial isopods, but they can survive underwater for several weeks, completely submerged, not even the option to get out of the water. So just something to keep an eye out for.
Yeah. Makes sense. Oh, I'm getting feedback. Here's a good question.
Work well with soil eaters. So that depends. In my experience, Dilatatus will just eventually become the dominant thing, more so even than something like Laevis or Scaber. And it just seems to be because they're generalists, and I think their niche is probably that they can tolerate drier conditions in the wild. You see them a lot on the West Coast in California, and they don't seem to have been established elsewhere. So, and I've seen them shred through even leaves that have not rotted completely through, even dry leaves. They will pick apart. It might take them, longer to sort of get to those like gigantic numbers, but I've seen them eat through cork bark too, and that's completely loaded with tannins. It takes some time, but they'll still do it. So something like Porcella dilatatus probably would not be keeping with other things because of that. The sheer shredding ability, and they'll ruin hides and other things that you would be keeping with other species, unless of course you've got a good gradient of humidity in there where the dilatatus will be able to pick their premium zone. The other thing will be able to get away from that or have a different zone that it prefers to be in. So
essentially you're creating that resource partitioning through microhabitat.
Mm-hmm. And that being said, because I'm crazy, I do actually have some Porcella dilatatus in a communal tank in my living room that also has Myoniscus ocellus, Stardust Dalmatian, I believe Armadillidium CF Espanola, I think they're calling it now. Mm-hmm. The marbleized. Yeah, Porcella duboscui, some double het Armadillidium nasatum, whiteout, and also orange. I feel like there's something, Armadillidium maculatum, a yellow stripes project that I've been working on for a while. So all those are in a big communal in my living room. Mm-hmm. And I put the dilatatus in there under the understanding that 2-3 years from adding them that I would have to go through and do manual extraction and rebalance the ratios in there. But other than that, the rest of the assemblage has been quite happy and quite productive like that. The duboscui are a recent addition. But they seem to be doing well.
Okay. So I really like what you said just now about having to go in and manually readjust the balance. Mm-hmm. I think that's something to keep in mind with these multi-species communals, that maybe people are just saying, well, if I can throw them together, I expect them to coexist indefinitely without maintenance. But that you're saying that that's not always the case. Mm-hmm. You may need indeed to... if you're going to do this, you're going to need to be willing to take steps you would not otherwise have to take if you were doing only one species.
Yes, and that depends entirely on what you add. If you had an enclosure of Armadillidium maculatum and Porcella scaber, I don't think that over a long period of time, as long as there's a very, 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 very slight moisture gradient, that you would ever have to go in and readjust the ratios with those. But if it's more moist, the scaber will do better. They probably won't completely outcompete the maculatum over time, And if it's drier, the maculatum will do better than the scaber over time. So it's just finding something that sort of accommodates both of them and not letting it swing to either extreme.
Mm-hmm. And providing, like you said, the gradients. Gradients are going to be key. Okay. Oh, here's an interesting question. I want to get your thoughts on this too.
I'd say unnecessary. Although, if you like the idea of using guano over, bat guano over using roach frass, then I can't really disagree. I would just, wonder about where the guano came from, and there's probably a reason why there aren't Cubaris or Nasodillo or actually any isopods in bat caves where bat guano is harvested. That is surprisingly one of those environments where cockroaches are exceptionally adapted, both in both places where there are a lot of bat caves, which would be South America and then Southeast Asia and parts of Australia. The dominant invertebrate in those communities is usually cockroaches. So I would think that there's no doubt in my mind that given millions of years, if you threw a couple of some sort of isopod into a bat cave with no competition, that you would get a very well adapted isopod to those conditions. But for whatever reason, they don't seem to do very well compared to something like cockroaches when feeding on bat guano. So it couldn't hurt to offer a little bit to see how they respond to it. What I can say from my personal experience that they are much more fond of is chicken poop, actually, and not the chicken poop that you can get in a bag of manure at, like Home Depot or something like that, but fresh, right out of the butt chicken poop, because it is just loaded with all kinds of bacteria, and it smells and it draws them in. When I go outside during the summertime to check on my chickens at night on the roosts and I look at the poop underneath them, it's just crawling with isopods. It's crawling with tons of things, but mostly isopods. There'll be some like spotted Mediterranean roaches, and there'll be some darkling beetles, but there'll be Porcellio spinicornis, there'll be Aniscus ocellus, there'll be Porcellionides prunosus, there'll be Porcellio scaber, there'll be Armadillidium nizatum, and Phylloseum muscorum will be on it too. And you can see all these species on just one little piece of chicken poop too. So I would recommend, if you have a bird, that might be a good option also if you don't have the time or whatever for chickens, or can't find somebody who's desperately trying to get rid of fresh chicken poop. And rabbit poop too. They don't seem quite as fond of rabbit poop, but once it's fermented a bit, again, I have a large compost, and to see the full assemblage of isopods out on that at night, which is quite impressive and a little terrifying. It's like, there's a lot of these here. So just the density numbers of them. Yeah. So,
well, that makes me want to try chicken poop with my isopods. I may well do that.
Yeah, give it a go.
Yeah. Cool. Well, another thing about bat guano that I think I may have mentioned somewhere else is that bat guano is not always sustainably harvested. There are times when it can negatively impact. It's basically poached from caves where people go in and do it in a way that's not safe for the bats. So that can be an issue. I'm not saying that's always an issue with bat guano, but it can be. So that's something too. When I was down in Arizona visiting with Peter from Bugs in Cyberspace, we talked to a bat researcher down there when we went to a bat cave, and she mentioned that people will get the bat guano for fertilizer and so on, but it's not always done in a way that is beneficial for the bat colony. So if you did use bat guano, you'd probably want to make sure it was sustainably harvested bat guano, or at least do your part to try to ensure that. So, all right. Let's see. I'm just kind of trying to catch up on all the chat. There's a lot of chat, which is great. Okay, there's a lot of people talking about the horse manure and the chicken manure.
Yeah.
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 It's great though. I'm really tempted to try it. So, and let's see. Oh, Chris says uses guinea pig poo.
Yeah, I also have guinea pigs that I raise outside, and at least during the summertime, and that's another good one. Just anything. It's really, the poop itself is digested, so it's not the poop is not nutritionally rich. It's the bacteria and all the gut flora that comes out of everything that they're really after. And then of course, once it's been sitting out and it continues to ferment out there, out in the open, depending on the moisture and blah, blah, it remains appealing or continues to get more and more tasty.
Mm-hmm. That makes sense. Oh, here's something Wally was mentioning about with the, if you're putting things like back guano in, you probably need to keep this in mind.
Nitrogen. Yeah, it is. That's why it's fertilizers, because of the nitrogen. And, it's interesting with the bat cave roaches of various sorts. They can be quite ravenous. Things like Eublabrus pycnoscelis, where this is a good point to plug this in there. Bat cave roaches bat cave adapted roaches do not make good communal species. They're used to living on top of other members of their species and eating whatever falls to the cave floor. And so eventually, you can have a very nice setup that's suited for Eublabrus or some of the pycnoscelis are bat cave species. But because of their incessant activity in the soil, just constantly disrupting it, making basically a moving carpet, and their ravenous appetites, they're not good for co-housing with any other invertebrates. So I think Eublabrus and a couple of the pycnoscelis. I want to say Phemapterus might fall into that. I'd have to look at some pictures again.
Cool. So that's a good thing to keep in mind. They have a specialized niche there, and so it's not necessarily going to match very well. So have you ever tried reptile poop with your isopods?
I have. Things like snakes. Snakes are very efficient digesters of their prey. I think having looked at, there was a figure I read a while ago on what carnivorous organisms have the highest rate of conversion of the protein and nutrients in their food to functional energy for themselves. What wasn't lost from them eating things, the energy that wasn't lost. I think the top one was something like minks or ermines or something like that. It was very high. It was over, I think, 90% of the energy from their prey they are able to sequester. And snakes are pretty up there too. It's something like somewhere between like 70 and 85%. So there's not that much left there. And another thing is if you're feeding things like mice, not pinky mice, but adult mice, the hair is not very digestible or palatable to a lot of organisms. And there are whole assemblages of organisms that are very adapted to eating mammalian hair and frass, like Trox beetles. Those are they're hair eaters, and they're very specialized for that. Beardy poop, which is has the reputation for being extremely smelly, stinky, is excellent, as you would imagine. Is very excellent fodder for invertebrates because of it's probably got a lot of bacteria colonizing it. So beardy poop is very good for different inverts.
I found that garter snake poo works pretty well, and garter snakes tend to be fast digesters. Maybe they don't digest as thoroughly as some other snakes. Their metabolism kind of pushes things through fast. So they do have kind of smelly poop, but my isopods love it.
Yeah, that's great. I think also with garters is the fact that they just sort of have a generalist physiology that, the trade-off there might have been certain enzymes for digesting the proteins and specific prey items has been switched out for more general purposes, considering they can eat fish and earthworms and rodents and amphibians and all kinds of stuff. There may that may have been the trade-off for being a generalist there, is not being able to, eating anything, but not being able to get everything out of anything that you eat, relative to other snakes.
Yeah, that's a good point. Okay, here's one from Christopher.
Oh, Aleniscus. So, key points for their care. I'll touch on that really quick. In my experience so far, not a communal isopod because of their specialized care, but we may find some beach-dwelling invertebrates that would do well with them. I'll actually bring the enclosure over because I keep it up here.
Cool.
This is going to be my arm workout for today. Oh. So this is the Aleniscus enclosure. It's a couple inches of sand from my yard that was sieved on top of a drainage layer of dolomite rocks with a little bit of frost cloth that stops the sand from going down into the rocks. And there's also a little port that allows me to pour water so it goes directly down to the base layer instead of having to drain through the sand to try and keep the salinity even throughout for as long as possible.
Oh, cool.
So the sand is moist. There's no filtration, no anything like that. Room temperature. And behaviorally, they're kind of like Hilaria, but a bit more active. You can get them out more reliably if you don't feed for a while, and then you do, or you feed a different food item. They'll come out in like a big surge, so that's pretty cool. But that's my enclosure for those. They breed at a fair speed. Definitely not as slowly as Hilaria. And for food, I found them to another thing as we talk about the importance of leaf litter and all this other stuff. They don't seem to be too keen on that, and I feed them mostly rabbit pellets and dog food and fish flakes. You really can't go wrong with fish flakes for a lot of different organisms if you're really desperate to find something will eat. So that's my setup for those.
Yeah, that makes sense. Fish food is one of my favorite foods. I usually use pellets, but it's basically the same idea. Oh, here's a question about the on the subject of poo. Have you tried blue tongue skink poo?
I've never had a blue tongue skink, so I've never been able to try blue tongue skink poo. I would imagine as long as if they're eating a diet of invertebrates, probably fine. The best way to find out some of these things to give it a try yourself and see what the results are.
Yeah. I bet Sandy Wood, she's a blue tongue skink breeder, and she also has isopods. So
cool.
You can let us know, Sandy, what you discover. And blue tongue skinks being omnivorous, they do eat a lot of invertebrates, but they'll also eat fruit and veggies and whatnot. So I think that would probably work pretty well.
Yeah, touching on that, for organisms that are adapted to eating poop, like dung beetles, things like the Phineas vindex, the rainbow dung beetle, and a lot of the other ones. People who have reared them in captivity, and my very sparing experience with those, and there's what's that one from Texas? Canthon imitator. It's another very generalist dung beetle. But from dung beetle breeders, usually omnivore poop is highly coveted by dung beetles. So so things like human dung, pig dung, beardy dung, etc. And I have raised Canthon imitator on beardy poop before. And I tried boa constrictor poop at the time. They didn't want anything to do with it. Again, the hair and whatever else is left over from digestion just was not very palatable to them.
Right. Oh, that makes a lot of sense. So omnivorous poop seems to be probably more nutritious. And like you said, with snakes, since they're so efficient, yeah. And then I've noticed that when I put isopods, when I put like an extra mouse, even an adult mouse in with my isopods, that the everything disappears. Everything is gone. Like the hair, everything. They eat it all. It seems to be. But if I put it with, say, my dairy cows, that's what happens. And not just pinkies. I do, throw extra pinkies, but even the hair on an adult mouse gone.
Jeez.
So
yeah, and it depends on the colony size. When you have a big colony of pretty much any species, and the good sort of that sort of momentum with the microbiotic community in the enclosure and in the individuals. With that momentum, a lot of things will just tear through anything. I would imagine my main rubber ducky colony would probably tear through a mouse if I put it in with them. So again, it depends on how many you have and what's basically the biological history of the culture.
Yeah, and that's something I love to talk about, this difference in a new culture, a moderately populated culture, and a fully established booming culture. Everything it's night and day. It's so different. And I think that's something that a lot of times beginning keepers don't realize. And they say, like, I have 12 zebra isopods and I haven't ever seen them. They're always hiding. And I say, just wait. It's totally different. As soon as they get going, you'll see them all the time. It's going to be
wait a couple of years. Or wait 1 year.
Yeah, exactly. Just once they get going, they're so different. Even things like dairy cows, which are probably one of the boldest isopods out there. When you first get them, a lot of times they'll just hide because they, like you say, the pheromones aren't set up yet. They don't feel comfortable yet.
And there's also, with a couple of isopods, they swap biological strategies depending on the density. So if you get 12 isopods and throw them into a 5-gallon tank, they will, most species, and it's most obvious with things like Armadillium vulgare, Porcello faster breeding things, temperate things, they will devote most of their energy into growth and becoming as large as possible and then producing as large of broods as possible. When they're crowded, when you take individuals from a crowded culture, they will begin reproducing younger and the brood size will be smaller. So this is another thing that factors in there, people getting isopods and setting them up in what could be an ideal enclosure, not getting any babies, waiting and waiting and waiting because the isopods, because of the density and the resource availability, their physiology has shifted to this grow big and produce lots of babies strategy over the, live young, breed a lot, die young strategy in a more established colony where there's, dozens of individuals.
Right. And that is part of the reason why people will see, people notice, okay, I got this new colony isopod and they're not breeding, but they don't know this reason behind that. And then suddenly, of course, boom, you have 300 in there because they devoted it to those large broods once they put on some size. I once found a monster zebra in my morning gecko enclosure, far exceeding any zebra I had ever seen because of that. Because there were very few in there, and it devoted all of its resources towards growth. Let's see. Oh, wow. Here is something about cohabitation. I like this question.
One earwig will pick off isopods here and there. It depends on the earwig species too. There are a couple that are fungivores, but they may opportunistically eat isopods. In general, not a good thing, especially if it's European earwigs. Those can eat isopods, especially freshly molted isopods. Stone centipedes will do that too. Occasionally, those will come in when people collect leaf litter outside. One stone centipede can inflict almighty hell on an isopod culture. Yeah. So, which is why another reason why I go through the lengths that I do to soak my leaf litter and all that is because I want to make sure there are no stone centipedes in there. So,
yeah, I've actually talked to several people with that issue, particularly having centipedes in their enclosures and how it decimates, especially if you get a gravid centipede in and then you get a bunch of little centipedes, and as soon as they hatch, springtails and isopod monki are the perfect food, and pretty soon you have a colony of centipedes and not so much So you've got some other critters there that we haven't seen yet, right?
I do. This for the Roach Communal live stream, really been going off on isopods, but I will let's dip back into the roaches then, shall we? And it all ties in together too. the Florida decided to go on a little trip there. There we go. So this is actually, it may look like just a regular, there we go, regular common hisser, but this is actually a female V-horn hisser. I think people are very surprised at how large these are.
That is a big roach, yeah.
And for being from the genus that has 3 very small hissing cockroach species in culture, Elliptorhina davidi, Elliptorhina chopardi, and Elliptorhina javanica. So hissers there's double-edged sword to hissers, and that is, on the bad side, hissers can cause problems for people who are hyperallergenic to insects. They produce a lot of saliva, they produce a lot of frass, they don't seem to digest some of their food fully, so it'll get moldy. So there'll be people who have problems with allergies to hissers because of how messy they are. Mm-hmm. On the flip side of things, those very same factors can make them excellent cohabitants with isopods because they're producing all of that stuff, and the isopods don't care. It's tons of high-quality food for them. Right. So keeping isopods with hissers is a great way to really turbo boost a colony. And I keep all kinds of species of isopods with my hissers. Porcellio bolivari, Porcellio nicholsi, Porcellio magnificus. I keep trying to say nasodillo, but I don't know if this species is in nasodillo or not now. The Cubaris species red edge, the orange mutation. They're just excellent because they're big, they're bulky, they tend to stay up in the driest areas of the enclosure. Their frass they can be so messy. So they're excellent for keeping with different types of isopods. And in fact, my entire Porcellio bolivari colony and my Porcellio magnificus, and in the past my Porcellio hasi. No, I still have one of the other lines of hasi in with hissers. They're all kept in exclusively with hissers. I've given up trying to culture them separately.
But they do great with the hissers.
Oh yeah, they do wonderful.
Yeah. See, that's something I wish I could do. I can't keep roaches, unfortunately. Not let me, but you've persuaded me already that would be an awesome thing to do. Mm-hmm. I do know that my Armadillidium klugi Montenegro, mm-hmm. I have a huge colony in with my garter snakes. That's they wonderful. Do exceptionally well in there, breeding mm-hmm. like crazy, eating I barely do anything to feed them because they're taking care mm-hmm. of the waste and the skins and everything, as well as the leaf litter that I put in there. Mm-hmm. It's fantastic. So I think with what you said, I might try setting up some of the larger Spanish Porcellio in another garter snake enclosure and see what I get. Maybe Magnificus or something like that.
Yeah, and when I did work with garter snakes, with eastern garter snakes before, flipping boards at the community organic garden to look for garter snakes, always be just tons of isopods under there. And there's a lot of people have a lot of worry because there have been some opinions spread around on different types of isopods being very ravenous. But again, it depends on your husbandry. Right. If you're keeping your isopods too dry, they're not getting the exact nutrients they need because you have dead, barely decomposed leaves in with them. They're gonna seek what they need to fulfill their dietary needs. And honestly, I did a little mini experiment once where I took I took an apple. And I took a piece of steak because somebody was very on about, Porcellio scaber, very protein hungry. And I said, I have this starving colony that lives in with my egg-eating snake because the egg-eating snake only gets fed once every 1 to 2 months. So I will take a piece of apple and a piece of steak, put them next to each other, give the enclosure a light mist, to encourage the isopods to come up, and we'll see what goes to which one more. And so I did this, and I took pictures of the colony for about once an hour. And at the end of things, the difference of isopods going to the apple versus the steak was not statistically significant. So I think over the course of that, maybe 100 isopods went to the piece of steak, and they were both the same size and placed equidistant from each other and everything. And I think maybe 80 isopods went to the apple. So which was not a statistically significant difference between the two of them. It could have been because the steak having an open bottom could be accessed from below, or the smell wafted further into the substrate or what have you. So In that case, I think they were more going after the moisture. The apple and the steak both have probably roughly equal amounts of moisture in them as well. So anyways, this was, you would think for something that was so villainized as protein hungry that I would see a glob of them on the steak, and within an hour it would be gone. Especially when they were only getting pooped on once a month by the egg-eating snake.
Yeah.
So,
that's a great point. I think, like you said, they can get vilified for their penchant for protein, but it's less. Not that they don't go after protein, but it's more about limiting factors in their environment.
Mm-hmm.
And if the limiting factor is moisture, then that's what they're going to get. And if the limiting factor is protein in a particular case, that's something else. But we just have to think of what are the limiting factors in this situation.
Mm-hmm.
So Aqua Gardens then has a question about Dubia roaches and isopods.
So wonderful, but it depends on how you keep your Dubia roaches. So people, the current factory farming for Dubia roaches is dry egg cartons, water crystals, pile of generalized powdered up roach diet in the corner, and that's that. And I can't even think of any isopods that can survive those sort of conditions. So those aren't going to work. But if you keep them, as I prefer and know is better for the roaches, with a substrate where the Dubia roaches can burrow, lots of bark pieces, a mixed diet, etc. Yeah, you can keep pretty much any isopods in with dubias and get good results, as long as you are not keeping them on a bare bottom enclosure with egg cartons and stuff like that.
Catering to the needs of the isopods for humidity and gradients and so on. I
mean, even the dubias, they prefer that. You'll see, I'm quite surprised when I see Dubia roaches for sale that they're usually, adult females are usually only about that big. And it's like the females in my colony, which is kind of like a traditional roach colony, with the substrate and everything, are about that big. It's pretty crazy, the night and day difference. And of course, you give it enough time, the populations that are in those, the factory farming of roaches, as hilarious as it is to say now compared to like 20 years ago, they will adapt over time, just like the roaches in the bat caves and whatnot did. But one of those compromises might be they're going to decrease in size as they deal with this very finite nutrient input and water conditions and stuff like that.
Yep,
so
selective pressure to stay small, breed fast.
Mm-hmm.
Yep, totally makes sense. Oh, here's another one about panchlora roaches.
Yeah, should work. A lot of panchloras are detritivores. And they tend to eat more of the substrate, and of course, whatever you feed, whatever roach food and whatnot you're giving them to. Some of the species, like actual Nivea, not the hobby species that was recently determined, have quite a penchant for rotting wood. And Prunosis, Porcellanides in general, tend to be leaf litter eaters. So, especially Porcellanides florea. Down south is found in sea grape leaf litter and whatnot. And of course, up here in Michigan, where I collected the first ones in the compost bins at U of M Dearborn, they were just eating the leaves and the grass clippings and stuff that people were dumping into the compost bin. You didn't really see them outside of the very heavily influenced by humans environment. So going into the woods with the oak leaves and whatnot like that, didn't really see them. But in the compost bin, that big slurry of different leafy organic matter, they would be there.
Cool. Here's another one.
Yeah, I actually the first Porcellanides prunosis whiteouts popped up from some individuals that got into my question mark roach, which is in the same genus as the domino roach. So yeah, you can keep them together. It's nice because the Porcellanides tend to stay on the surface, or just just barely below the surface of the substrate. And the domino roaches, although the nymphs are burrowers, they aren't actively churning up the substrate constantly. They just tend to sit and wait underneath the substrate until whatever causes them to move. So they're not very disruptive. So that's a pretty good combination.
Cool. All right. Let's see. Here's another one. Have you tried edible seaweeds with isopods?
I have. That was a go-to that I had when I was trying to find a reliable food for, what are those big coastal Ligia species?
Oh, like the Palaci?
Yeah, these were Bradburiana, I think, from South Florida. But the thing with the seaweed is it's salty, and the coastal species are adapted to that, but they didn't seem to prefer it over the fish flakes that I was giving them. And so also because of that salt, I don't think it would be the best idea to give it to strictly terrestrial isopod species. You may mess with the salinity of the enclosure, which not all isopods are adapted to increased salinities and whatnot.
That makes sense. Oh, we got a super chat from Mr. and Mrs. Morelia. Thank you.
Saying bioactive feeder colonies. I would be down in the future. Yeah.
Oh, yeah. Yeah. Can we plan on that in the future sometime?
Let's do it. There was another thing I was going to get into with this is with a feeder colony, you can still produce a ton of one species or whatever and still have it be a communal enclosure. One case in point is there was a time where I did not sterilize my leaf litter properly. I was adding it to my hissers and whatnot like that. And I ended up infesting a lot of my roach colonies with little unimobius ground crickets from outside, out my yard where I collected the leaf litter. Mm-hmm. And so now I have a bunch of roach bins that just have these huge colonies of unimobius crickets in them. And they get along fine, as long as they seem to really hammer the dog food that I put in and ignore pretty much everything else. And, there's always more dog food to go around, so there's never any problems with the roaches or the isopods or whatever else is in the container. Cool. So, I would love to do a video on doing some bioactive feeder colonies and whatnot.
Okay. Well, we have our next topic for our next video already then.
Yeah. Wonderful.
Cool.
And this can be extrapolated. I've touched on a lot of pet roaches and pet isopods and whatnot, but you can extrapolate from there that, as long as you have that sort of resource and spatial partitioning and you're aware of the needs of whatever species you're combining, you can get some good production out of a lot of these species, by doing in a communal setup like that.
Yeah. Yeah. Well, it's funny what I've got going. You mentioned the Uricotis that I had going a couple of feet from me. I have a bioactive leopard gecko enclosure. I've got Uricotis sigillatus breeding in there. I've got Zoophobus morio breeding in there. I've got Tenebrio molitor breeding in there, and I've got Porcelaneoides pruinosus breeding in there. All breeding in there. And the gecko does pick off some of them, but the populations are still maintaining because I add extra food for them. So food's not a limiting factor. And so I do, of course, feed the leopard gecko other things, but they're going in there. And whenever I want to breed more superworms, I just pull beetles out of here because they pupate like crazy in this substrate with the bioactive substrate for the leopard gecko.
Because of the depth, and they can get away from each other, so they don't have that. I have a little friend here.
So yeah, I think there's so much room for this kind of thing. That's like largely undiscussed. Like they're saying, like Mr. and Mrs. Murley were saying on YouTube, it's not that common, but there's some great room for all this kind of thing.
Yeah, and it's nice to be able to tie together the practicality of keeping things you want to feed your reptiles or what have you, and also being able to keep something that's that you really want to keep, something like isopods or specific type of cockroach or something like that, where you can save space and you can accommodate the needs of everything without putting in extra effort or food or anything like that. It's nice to be able to create little, basically mini ecosystems. And of course, you're putting the input in, which is the food or the water and whatnot. And that's all you got to do.
Yeah, and it's always fascinating me. I've been doing it for years in various forms. I've got the aquatic crustaceans. I've got Daphnia and scuds together, for example, and I've been keeping them like that for years. And you put the food in for the scuds, they eat it and they particulate some of that food, and the Daphnia eat that, and they both breed like crazy. You get two different live foods for the price of one. So it's the same kind of thing. Same kind of thing. Oh, who's this guy you got on your hand now?
This is Archimandrita Teslata. It's a peppered roach. And this is the roach that I keep in with that whole slurry of isopods I had discussed earlier, the big communal tank in my living room.
Okay.
So huge, handleable cockroach. The frass pellets are huge and nice and goopy for the isopods and whatnot. So another thing with going with roaches and whatnot is if you can keep roaches and isopods together, they're roughly the same size. The isopods I found, as long as the humidity is good enough, tend to be the victor in that combination for whatever reason. So I would not keep like Porcellio Hoffmannseggi with something like African bullet roaches or something smaller like that. But you can scale the nice thing is you can scale the roaches up a lot larger than you can the isopods. So you can
Definitely.
Blaberus and Bersatria and Archimandrita with a whole slew of different smaller isopods, and they still do fine together.
Mm-hmm. Wow, that's awesome. Someday I'll figure out a way to do it. Oh, that's so cool.
And I should also add, there's a lot of different detritivores work for this too. Roaches, I'm very familiar with roaches. And I have a lot of roach bins, so that sort of facilitates that. But also millipedes, and it makes that one makes even more direct sense. Millipedes and isopods are often found in direct association with each other in the wild. Not just the big ones, but flip a board again, you'll see all the little introduced julids from Europe mixed with all the different introduced isopods. So the larger millipedes can also facilitate the growth of the isopods. They break apart the food. They have the big frass pieces and whatnot. The only thing to watch there is not that the isopods will go after the millipedes or vice versa, but making sure that the substrate doesn't get completely exhausted as you watch the numbers grow, because at some point it'll end up just a sea of millipede frass, and there will eventually come a point where you need to refresh or add more or whatever, but just sort of keeping an eye on that. So I have things like my Centrobolus splendidus, the red fire millipedes, ones that are solid red. And somebody was saying like, oh, how do you keep these? Like, oh, I have them in this tank. There's a bunch of wood and leaf litter and a huge colony of Porcellio scaber. And like, I could feel through the text, they felt like their face went like white, like they went faint, like scaber, like don't they even know the millipedes burrow when they need to molt? And even then, it sent him a picture, and it's just a sea of Porcellio scaber and all these little red specks of the Centrobolus poking up from underneath them. It was fine, I had a pile of dog food everybody could go to, plenty of wood and leaves and whatnot. The scaber population just kept growing until I decided I want this whole colony to look like this one and this one. And then I reset it. Now we let the genetic dice roll a couple hundred more times, and we'll see how things go.
So in that case, do you have any issues with the millipedes? I know that millipedes, when they protect their eggs with a kind of a pellet around it, that can help keep the isopods off. Has that been your experience with breeding?
That seems to be the case, at least with Centrobolus and Tylobolus. Do I have a Narcissus? I have a Narcissus culture that I keep Porcellio silvestrii in. So that hasn't been a problem there either. But it depends on the millipedes, I suppose. So a lot of the larger, like spirobolids, are probably fine. I can't speak for things like Oxidus gracilis or Helicothermomorpha horstii, the weird black and orange ones from Florida, the flat millipedes and whatnot. So I can't speak for those since I don't have enough experience yet. But in general, as long as there's enough food of all sorts, everybody gets along just fine.
And that's a key point that has come up a couple of times. As soon as you reach a point where there's a limiting factor in the environment, that's when the stresses start coming in and the difficulties start coming in. Yep. And it can be a fast transition. You can get to the point where, suddenly the leaf litter exhausted. I didn't notice. Boom.
Yeah.
You can have some issues. So you have to keep on top of it. Yeah. So that makes sense.
Which is, it's also interesting because For these things, keep on top of it is, add a handful of leaves every 2-3 months, and it's interesting how everybody from, smaller keepers up to people who have large collections like myself, where it's like, we consider adding a handful of leaves or rotting wood every 2 to 3 months to be like, oh, got to keep up on, like something you have to constantly have in the back of your mind, like, oh, I have to go home and do this, It's just interesting versus, keeping different types of herps, something like a lizard where it's like, oh, I need to feed it every couple days or something like that, or keep a direct eye on the needs, something. It's interesting we kind of conflate the two of them. It's like, oh, 2 months from now I got to add a handful of leaves in here. I got to like prepare for that,
Yeah, well, that's a good point, because with invertebrate care, often it's so minimal with it. And it does depend, those of us who have larger collections, like I have a moderate collection. Yours is larger than mine, but when you have just my isopods, I have about 80 bins. And so if I were to put a handful of leaf litter in a bin every day, I might not be keeping on top of it if I only did one a day. So yeah, but I totally see your point. It's true. We're spoiled to that extent where minimal maintenance is pretty minimal, and it can be. Okay, well, we've got a couple more questions here. I think I don't want to step on Wally's live stream that's coming up soon, but I do want to cover some more of these questions. Can you tell us about this one?
So the Sylvestri I have currently are housed with Narcissus americanus locality. It's just rotting leaf litter and rotting white rot wood, along with some what is called flake soil. It's not necessarily soil. It's just basically composted wood pellets that have been colonized by fungus mycelium. And I found they do seem to do well cooler. Unlike, and this seems to be another big thing, is people going back and forth whether or not a species like say cold or warm or whatever. I remember there were some reports of Porcellio bolivari when they were first being cultured, where people were saying, oh, if they go over 68 degrees Fahrenheit, they'll all die. And then there was me sitting here with the bin of hissing cockroaches with the heat lamp on them, just overflowing with Porcellio bolivari. I was like, yeah, so they do seem to like it cooler. I've actually very successfully co-housed them with Doralea orini, Oren's zebra roach. The two of them seem to like similar conditions, sort of an even consistency of moisture, and just a little on the cooler side. Like I would not be adding much supplemental heat to Doralea orini at room temperature. And off topic, lumber grasshoppers do make sort of a rasping sound. It's not like a cricket chirp. It's more like you're shuffling through some papers or something. And that's the males when they're on the females courting them. They'll ruffle their wings like that and sort of lurch back and forth. It's quite humorous to watch.
Let's see. There was another one about roaches I wanted to catch here. It was about there it is.
Dryer needing roaches. This is actually something I've been eyeballing a lot lately, and I have some good results there. So Armadillo officinalis, very dry adapted. Most of the Armadillidium are good for, as long as there's a moist corner, they're good. Especially Punta Cana seems to have exceptional dryness tolerance. I'm currently working on a very dry tolerant line of Porcellio scaber. It's been about 2-3 years in the making. And they so far can get down to the point where it's almost, if the room humidity is about 40-50%, the whole substrate can be dry except for just the slightest touch of moisture in one corner, and they're still okay. So that's something I'm just going to keep, subjecting them to that pressure over and over again, and maybe in about 3 more generations, release it with an asterisk that, oh, these are the attributes that they seem to possess. But innately, Armadillo seem to be very dry tolerant. Armadillidium, except for Nizatum. Nizatum seem to be pretty dependent on moisture. And Porcellio ornatus, but not the dotted ones, or I think those were originally called South. The ones with the 4 yellow dots. Not those, but the high yellow and the witch's brew and whatnot. Those ones, very, very, very good dryness tolerance. Again, I've actually currently, I have been finding Porcellio ornatus evenly distributed throughout my Uricotis and Procera enclosure, where half of it's pretty much completely dry and the other half is moist to damp. And they will be evenly distributed throughout the enclosure substrate, despite the fact that the room humidity is only about 50% right now, which things that sit out in 50% room humidity, even with dryness adaptations, tend not to last very long. Even with the airflow and whatnot in the bug room. Oh. So yeah, Porcellio ornatus, high yellow, whatever other line, chocolate I think was derived from those as well. Mm-hmm. So that would be another one to look at. But the witch's brew as well.
Cool. I've noticed that my yellow dots are pretty dry tolerant. I don't know if it's a different line, or I've heard there's actually a cryptic species going on too.
Yeah,
so.
I really suspect that to be the case. The morphology between, or somebody misidentified or something, the morphology between the those dotted individuals and the high yellows and whatnot is very, very different. Mm-hmm. So I think there's something more to be explored there.
I think so too. Okay, this should probably be our last question. I don't want to step on Wally's live stream, so let's.
All righty. Hello, some eggs. Following me between live streams, I see. Any go-to combinations for roaches that do well together? I feel like this is a good question. Usually, small ectobiids can be kept with pretty much any larger roaches. So you keep things like Parcobletta, Echinoptera, Pseudomops, Hemithyrsospora can all be kept with things like Blabarus, all the hissers, and things like that. Because just the sheer size difference, they're not going to be directly interacting with each other. And roaches are so alert and whatnot, you don't have to worry about, is it going to step on them? Are they going to crush them or something? They just naturally avoid things that are sort of getting in their way like that. So a lot of the small ectobiids can be housed with the larger roaches. Things like banana roaches can be housed with things like Uricotis. Again, roaches that have very different preferences for where they want to be in the moment are good for co-housing. I had talked to you before, Russ, about I really wanted to get a picture for this, but unfortunately could not. It had to be an organic experience to find it. But I've currently been keeping Bantua robusta Namibia with Corsonura parishii. So it's a dry tolerant relative of the roly-poly roaches. It's about that large, with Corsonura parishii, which is about that large as an adult. They do exceptionally well together. So basically picking things that are different sizes and that have relatively similar humidity and whatnot needs. And even then, if you have a humidity gradient in the enclosure, things will sort out where they want to be, which is one of the reasons why the ectobiids do so well. A lot of the smaller roaches, is because a lot of them, although they are prone to desiccating, they also really don't like to be stuffy or too humid. They will, for whatever reason, their physiology is not adapted to that. They're used to moving up and down in the leaf litter or physically relocating if things get too moist or dry. So when you put them in a small enclosure, they aren't able to do that per se. So keeping them in a larger enclosure with larger roaches is a great way of being able to culture them without accommodating their picky needs in a smaller enclosure, and also having a mixture of different roaches together.
Cool. Best of both worlds, really.
Mm-hmm.
Cool. Well, thank you so much, Kyle. We've got to wrap up now, but it's been great, and I've learned a lot and had a lot of fun. So thank you.
Wonderful. Thank you again for having me. And I guess I'll see you in the near future for another round of discussions. So.
Yes, some of those bioactive feeder enclosures.